Okay, so I'm doing a reverse transcription. So that's to reverse transcribe or change my RNA that I've extracted from urine samples from patients into cDNA so that we can measure these things called microRNAs in their urine. So what I'm looking to do is to measure the microRNA changes in patients with diabetes. The aim of that is to predict whether these microRNAs change to better predict whether these diabetic patients will go on to get diabetic kidney disease.
So the project that I'm doing is mainly looking at what we'll be able to apply to transplant kidneys. So we're looking at the injury that the kidneys get in that time when they don't have blood supply, and basically looking at ways that we can reduce that so we can improve the outcomes of kidney transplants. So I've got here kidney tissue, which we've basically dissolved in an agent called Trizol. And the plan for today is to extract the RNA from it, so basically the genetic material. So this is the starting point, but just because I've defrosted them at the moment, I'm just mixing them up basically. Basically now I'm using the centrifuge to spin the liquid in the epidermis. So by the time it gets to the end of this programme, it will be split into its different components, and the RNA will be nicely sitting at the top, so then I can take that off, and then all the experiments from here on will just use that RNA and get rid of all the waste.
Right, so currently I'm working on a research project that is looking at a specific receptor involved in fibrosis. So we know that it's involved in fibrosis, but we want to know how. And when we know how, then we'll be able to prevent it. So fibrosis is, in effect, scarring of soft tissues like organs such as liver, kidneys, but it can also be seen in the skin, so excessive scarring. So when you have a cell known as a myofibroblast consistently present, it lays down extracellular matrix, which is involved in the scarring process. Here I'm making contraction gels, and I'm treating the cells known as fibroblasts with TGF-beta. When treating them with TGF-beta, they become these myofibroblasts that I spoke about earlier that are involved in fibrosis. So I'm looking at putting them on these collagen gels and looking at the contraction that results from these myofibroblasts being present. The next stage then is to silence the receptor that I think is involved in fibrosis and see whether it reduces this contraction. Right, so this is how I'm making the contraction gels. So we use something called collagen, which is present in your skin in abundance. Mix that with some medium, and then we have to make it to the correct pH so that it doesn't kill the cells. So just add this is just medium that makes the cells happy. It gives the cells these nutrients that are required for them to grow happily. Knowing more about it will allow us to understand it better. With a greater understanding, then we can combat it better.
Okay, at the moment what I'm doing is I'm looking at how different genes are expressed in a particular disease state. What we want to do is be able to stratify disease to better inform treatment for different diseases. So we use a technique called quantitative polymerase chain reaction. So I have samples from a disease model, and then I use this PCR reaction to amplify the genes and quantify them. So this particular model I'm interested in is a model of arthritis. Arthritis treatment is very good, but the disease is very heterogeneous. And we want to be able to better target the drugs to particular diseases, so different types of disease. And using this method we can pick up gene signatures, so we can see which genes are dysregulated in particular types of arthritis, and that allows us then to use the drugs more effectively.
I'm Bob Stenman, I'm the Outreach and Engagement Lead for the Wales Kidney Research Unit. From my point of view, it's very important because we need to get out there and tell patients, get the patients more involved, tell them what we're doing and find out more about what they'd like us to be doing.
lab staff are doing research into all aspects of renal disease, and we just help and facilitate from the admin point of view.
Donald Fraser, I'm the director of WKRU, and I suppose my roles are, first of all, in terms of running a programme of research that's my own interests, if you like, around microRNAs and their use as biomarkers in the kidney, understanding what's going on in somebody with kidney problems non-invasively. And Tim Bowen here is a non-clinical academic who's the other expert who runs that group with me. So we have that work going on. I also have a role in terms of clinical trials on the clinical research facility, and then as overall director I try to keep things together and make it the best environment possible for people to come in and do work that's relevant to kidney patients. As to what I'm doing right now, tearing my hair out on a grant application, which is a common feature of what we do, just desperately trying to get it all in place in time for the close of that grant. We'll see whether it's successful or not, and Leah, you've been involved in that one already. So yeah, we're trying to get, as you know, trying to get this one in and take on a new piece of work that we've already talked to patients about, and we'll see if we can get that off the ground next year or not. So I think that it benefits patients. Well, I think there's multiple strands. The first of which is in the very long term, everything we do, we're hoping, is going to mean changes in patient care eventually. Some of the work which you probably, I don't know, have seen down in the lab can be quite a long way from that in terms of its identifying the very first mechanisms which then might lead to a change in therapy, and that can be many years from the first experiment on the bench, if you like. But some of it's much closer than that, because we're taking samples from clinical trials, for example, and saying, in this person who's being treated with a new type of treatment, what's happening? You know, can we find out something about them that allows us to measure responses to treatments a lot quicker than what we do at the moment, which is measure changes of kidney function over several years. So there's the kind of direct aspect of the work, benefits patients. We hope, you know, all of the different types of work that we have going on. The other thing is, I hope that through our outreach work, and you've been a sort of, you know, a massive contributor to that yourself, Leah, that are informing people about what's going on here and about kidney work more generally maybe has its own benefits in terms of the general public and people interested in kidney problems. And we've had quite a few events recently of patients coming in and you know, other clinicians or clinical staff of varying types coming and understanding what goes on in a research laboratory.
I'm a non-clinical academic who's working here. My principal interest is in gene expression, which used to mean DNA sequences in the genome which encode proteins, but now we know that other molecules called RNA molecules are important also important in regulating gene expression, and principally my interest is in gene expression in the context of kidney disease, and that involves working on small molecules called microRNAs. We're looking at those specifically at the moment as urinary microRNAs as biomarkers or sentinels of kidney disease. And one of the advantages of looking at urine is it doesn't require taking invasive samples like blood samples, kidney biopsies. And I've also been involved in work in extracellular matrix, which you've heard about from other researchers here.
So my project is looking at the immune response of peritoneal dialysis patients, especially when they get a peritonitis infection. So I'm looking at specific biomarkers so we can identify the infection quickly, and then we can hopefully treat it more effectively. I'm currently in the final year of my PhD, so I'm writing up my project with the aim of publishing it soon, and hopefully going to move on to something similar. So what we're working on is we're working towards diagnostic tests to be able to identify the infecting organism nice and quickly.
I have been growing up some kidney cells in the lab, and I've been tinkering about with them and knocking out a gene that I think might be important in diabetic kidney disease, which is what I'm interested in. So now I'm just having a look at the proteins in those cells, and I'm putting this red dye on to see if I've got any protein here. Every experiment I do about 8 of these blots, because I'm looking at a different So you either overexpress, or so you turn up the gene, or you knock it down, and then what I do is see how that affects the cell in terms of how it can respond to insulin, which is obviously important in diabetic kidney disease. So to do that, I need to look at a number of insulin signaling molecules, so the insulin receptor and other things like that. So each of these individual blots I'm staining for a different protein. Right. But this is purely just to see whether I've managed to capture the protein on this bit of membrane, because if I haven't got it there, there's not much point in staining it for anything else. Nope. Should we keep going until I find it?
Welcome to science.
This is science. There is actually some protein on this one, right at the bottom. There's like a little smear of darker. Can you see that? So this is the ladder. This bit here is the ladder that tells me how big my proteins are. And then the proteins appear and I can see which protein it is because I know what the molecular size of it based on that ladder is. And can you see there's like one there, one there, one there? So that is a bit of protein developing. But that explains why I'm not getting very good results from these plots. So probably back to the drawing board for this one. Well, hopefully what I'm trying to do is look for a new marker of diabetic kidney disease. So this could be useful for therapeutics, so something that you can target to control the levels of that might improve your responses in diabetic kidney disease. Or it could be something that we use as like a biomarker, so something that might be in patients' urine or blood, for example, that we could use to detect diabetic kidney disease at an earlier stage than we can at the moment. So quite a few potentials for it, but you know, at early stages at the moment.
These things that I'm taking out here are my screen printed disposable electrodes, and basically what they're in at the minute is a solution of phosphorus pentachloride and ether, which is why it doesn't smell very nice, and they're essentially modifying the surface so that I can attach DNA to the surface, which is what I'm going to be doing in a second. So there's in my little sample tubes on the side there little sequences of DNA which I'm trying to attach to my electrodes so that I can analyse samples of another thing called microRNA which my DNA attaches to. So I put the DNA on here, I then run a scan over there and the microRNA attaches to the DNA and then it changes the voltage. The idea being that these things can be used with urine, and when you have in kidney disease in urine you have different levels of the RNA, and then if we can quantify the RNA we can tell you if you've got kidney disease. So the overall aim is, because at the minute if you get kidney disease you sort of progress onto needed transplants or dialysis and that sort of thing, and the minute they diagnose it through proteinuria and that sort of caper, but by the time you've got the proteinuria it's kind of too late. So we're trying to develop these electrodes as first means tests that can be done at the GP, so that you don't have to worry about progressing on to, we can treat you medically if you like, rather than you having to have blood tests and the other one where they take a bit out of you, but I forget the word of. Yeah, biopsy, thank you. So you don't have to have blood tests and biopsies, you can just literally take this electrode, dunk it into your urine, it will tell you whether or not you've got kidney disease. Obviously the idea is giving the patient the best quality of life that we can. We don't want to have them having biopsies or blood tests if they don't need it. And obviously the earlier we find it, the less likely you are to progress onto needing dialysis and particularly harsh treatments later on. So, I mean, it's in the early stages yet, but hopefully. So before before I can put my DNA on my electrodes, that's all my electrodes come like in the card. Before I can put them, I can put the DNA on there, I have to modify the surface. To do that, I'm currently using a diazosulfonic acid, but I have to sort of do some chemical synthesis to turn it into that. So this is what I start with. This is the pink mush. That's just water in there, it's just dissolved it. Basically add it to that, and if you have a look at the top, it's called sodium nitrite. It will turn one of the amine groups in there into diazofunctionality, which is basically two nitrogens attached to each other. and over time, so I'm just adding a bit of acid, it just sort of speeds the reaction up. You will start to see it change colour. So that orangey colour indicates that a diazo group is a charge group, which means that it reflects light of a different wavelength. So the colour change is indicative that I've got that diazo functionality. Once I've done that, once I've added it to the electrode, I have to run the scans on it. And to do that, I've got a setup over here. And it's not particularly the most rudimentary setup, so I apologise for that. But what I do is I essentially put on an electrolyte. It's called ferrocyanide. It's got cyanide in the name, but trust me, it's perfectly safe. I am a chemist, so I would say that. And I basically just apply that to the electrode. Basically what I do is I apply a voltage to those electrodes, and then those voltage generates a current. The more current that I get, the more microRNA are on the surface. This is just a clean one, so I'll just show you what it looks like basically. But it's not the most exciting thing. This is what I have to look at every day. But basically a voltage will be applied, and as the voltage applies, after a couple of seconds, so you see how a voltage is applied. As voltage applies, a current is red until it reaches a peak maximum, and then that's where the electrolyte is oxidized, and then it will come back the other way and apply a negative voltage, which will cause it to reduce. And then the bigger these peaks are, the more RNA is on my surface. I mean, what I just did there would be what you do in the doctor's office, if you like. They shouldn't plug it in. I mean, ideally they dunk that into a solution of this stuff. I'm just doing it quickly, but you plug it in, dunk it in, press go, and then the readout will automatically tell you whether or not you've got kidney disease and that sort of thing. And in the time that I've taken to talk about this, hopefully my little reaction will have got about over here, and it'll be a nice different solution, which it is, hey! So you can see, or I've actually done it properly now. Chemistry, that is, on the go in the biology lab. Can you view it? So all I do with this is I simply attach it to the, with a pipette, same on my electrodes again, run an electric current through it, and that causes the molecules that are in here to stick to my electrode, and then with those molecules there I can then attach that, the DNA to that directly.
So I collect and analyse all data for all whales on the acute kidney injury electronic alerts. And then on a quarterly basis then health boards can get reports on their incidents, their outcomes of AKI. The data also supports quality improvement projects throughout the health board across all different hospitals in Wales. So the main aim of the data is to provide support to quality improvement so they can improve the response to patients, which ultimately improves the patient outcomes for patients with AKI.
This is the tissue bank. So we are in the freezing facility for the Wales Kidney Research Tissue Bank, which is a collection of samples donated by patients and healthy volunteers. So we have a range of various types of samples with mainly blood, urine, peritoneal dialysis effluent, kidney biopsies, which are kept somewhere else in the pathology lab of the hospital. But here we have all the other type of tissue which are ready to be transferred to researchers when they apply to the tissue bank. This is where we store the samples, so they're all organized in rows and boxes so that we can find them easily, and everything is logged into a database where we have the history of all the samples, when they are collected, and what has been done with them, and who we are transferring them to. So that's what it looks like.
Okay, so I'm taking samples out of the centrifuge. Basically what I've done is, these are cells from kidneys. And what I've done is I've put them in triazole, I've dissolved all the cell components, and the RNA, I don't know whether you can see the top. So the RNA is the clear liquid at the top, and that's what I'm going to take off the sample. And I'm going to leave the pink stuff because that's all the DNA and all the other cellular stuff. So basically it just involves trying to be really careful and taking off the top layer and leaving the middle layer and the bottom layer. Don't know how well you can see that. So I'm just taking that layer off and putting it into a new tube. So that lays the RNA, and after that I'm going to purify the RNA a little bit more, and then that will go on for analysis to see what microRNAs are in there. So like I said, these are from kidney cells that I've grown up, and I've treated them with glucose and TNF-alpha, so they're both things that you'd find in a sort of diabetic kidney. So I'm going to see how that influences the microRNAs that they send out, basically. Basically what we're trying to do is sort of an artificial model of what happens in a diabetic kidney. So essentially if we find out something that these cells do in response to high glucose or high inflammatory mediators, that could potentially inform new treatments in the future, and just gives us a better idea generally of what goes on